As of 2023, India had the second largest global burden of the fatal and genetic sickle cell disease. Current methods pose various challenges when it comes to large population-wide screening, which is a critical stage for the national elimination mission. A group of biophysicists at RRI have developed a low-cost, high-throughput device for the detection of sickle cell disease, optimized to be available at any point of care.
The idea is to understand how stiffness of a biological cell can be used as a biomarker for early detection of disease.
For this, we use a tool which is called a resistive pulse technique.
In this method, we take a microchannel filled with electrolyte solution and we apply a voltage across it to get a baseline current. Whenever any particle passes through this microchannel, it causes a current drop, which tells us about the size of the particle. Because bigger particle will cause more current drop as compared to the smaller particle. To get the stiffness information about a biological cell, we use a microchannel whose diameter is smaller than the cell size. So the cell must squeeze through the channel, and here the rigid cell will take longer time to pass as compared to the softer cells.
The height of this can be mapped to the cell size and the width of these cells can be mapped to the stiffness of the cell.
We treated RBC with a drug latte that softened the cell and measured the stiffness by AFM. This helped us calibrate our device to distinguish between cells having different stiffnesses. Then we use our device to screen sickle cell disease patients based on their red blood cell stiffness. And we can clearly distinguish between two populations of sickle cell RBC and healthy RBCs.
The next step is to expand the testing with samples across India and across ages to ensure the reliability of the method. And if all goes well, this may be a huge factor in speeding up the screening step of the National Sickle Cell Anemia Elimination Mission.